All right. So today we continue learning about nearshore environments, and we go back to learning about algae. We had a talk about algae about 2 weeks ago. The guys from the Eva Beach Limu project came out, and so today we learn about the phylogeography of tropical Pacific marine algae by Dr. Alison Sherwood from the University of Hawaii. So we can give her a round of applause.
Thanks very much, Mahialani. I appreciate it. It's my pleasure to be here today to speak to you about some of the work that we've been doing in the lab and bringing that together with what we know from the literature on the evolutionary patterns that we see within species of seaweeds in the tropical Pacific. So we know, living here in Hawaii, that it's a pretty special place, and organisms that have evolved here are particularly special because they've had to arrive by long distance dispersal. We know that this is an extremely isolated group of islands. And what it means is that the organisms that we find living in freshwater and terrestrial areas in the islands are shaped by processes like adaptive radiation, where a single colonist would arrive in the islands and over time would evolve into a whole series of different, very, very unique and closely related species. The marine organisms are a little bit different. So we see different patterns for those organisms a lot of the time because there's a greater degree of connectivity in the environment for the marine organism. Just by virtue of them living in an ocean that is connected, we see different evolutionary patterns. So the species that we see here in Hawaii are shaped by many factors. They're shaped by things such as having arrived from a long distance away. They're shaped by the different habitats in which they live. They're shaped by their different life history patterns. And all of these are reflected in the patterns that I'm going to be presenting to you today. I want to start off with the definition of a word that's in my title, phylogeography. Phylogeography is a science where we can look at the distribution of gene lineages within species, and you can use this to interpret the evolutionary histories of species with respect to historical events. So it's bringing together what's happening in terms of geological history of the Earth with what we find going on in the evolutionary history of certain organisms. These kinds of techniques that we use are very useful, and we want to develop hypotheses of where organisms originated, so where they came from, how and when they arrived in new locations. So it's a useful method for combining geology, biogeography, and evolutionary biology. Okay, well, we're talking about the tropics today, so I'm going to spend a little bit of time talking about, in particular, the ocean basin that we are in right now, the Pacific Basin. Let's start off, though, with making sure we're all on the same page with respect to the tropics. So we all know this, that the tropics refers to the area between the Tropic of Cancer and the Tropic of Capricorn. It coincides approximately with the hermatypic coral distribution, and it spans 3 different ocean basins. So this is a swath or a band of ocean which stretches around the Earth, including the Atlantic Ocean, the Indian Ocean, and the Pacific Ocean. So a long time ago, approximately 200 million years ago, there was a very large ocean that developed when Pangaea split apart into Laurasia and Gondwana, these 2 very large land masses. So the Tethys Ocean was the name of this ocean. And it was so large, spreading around this tropical region of the Earth, that what it did was enable the evolution of a whole series of organisms that had pantropical distributions. Okay, so the Tethys yielded pantropical marine species. And as the continents continued to move around, these subsequently became isolated. And you can see the remnants of this pantropical distribution in many organisms today. The Pacific Ocean, where we live, inhabiting the Hawaiian Islands, came into play a little bit later. After the breakup of Pangaea, we start to see the Pacific Ocean forming. It was heavily influenced, though, by the Tethys, and so were the organisms that lived in the Pacific Ocean. The Pacific is a really interesting place to study phylogeographic patterns. This is a very, very large and very old ocean basin. which means that there's been a long period of time for organisms to evolve here. In addition, the distances between islands or different kinds of habitats around the ocean basin are very, very large. And this can come into play when you start to think about how organisms become isolated and start to evolve into different lineages. One of the really interesting things that I think has probably shaped the evolution of marine organisms in the Pacific Ocean is the fact that this basin is characterized by many island groups. And what these island groups do is break up the dispersal distances. Okay, so it means that there's a shorter distance between the different groups that organisms would need to disperse in order to colonize. And it provides opportunities as well for partial isolation of populations. These islands are also really interesting because they have come about by a whole series of different mechanisms. So they are not the same in terms of what they're made of and how they developed. And those factors have also shaped the evolution of the organisms that we see in the ocean basin today. So here are some examples. Here in the Hawaiian Islands, we know these are volcanic in origin, so they came about through hotspots. There are a whole series of islands that are characterized as seamounts. An example of this would be the Line Islands. Some are atolls, and there's a whole series of these around the Pacific. The Cook Islands, or some of the Cook Islands, are a good example. Some of the islands are in fact uplifted reefs or atolls, and some of the Solomon Islands qualify in this category. And then there are some that are essentially fragments of continental crust. That is, they are not oceanic in origin at all, but broke off of much larger land masses. Places like New Zealand and New Caledonia. And you can imagine that this has probably had a really large effect on the kinds of organisms that evolved there. If you have a piece of land that has broken off a larger continent, it's bringing with it, in theory, a series of evolutionary lineages. Okay, so when we think about the process of speciation in the marine tropical Pacific, a lot of the historical literature that you can read about has been based on an assumption that these marine species have had planktonic larval stages that disperse well, either by short, medium, or large distances, but nonetheless that there's some kind of readily dispersible unit in the life history of these organisms. What I'll be talking about today, though, in terms of seaweeds and their life histories will be a little bit different from this. And some of the really charismatic organisms that have been studied include things like the yellow tang, green sea turtles, and limpets, opihi. We know that population connectivity decreases with increasing distances, and there's been an assumption that this leads to greater isolation and eventually speciation. And corollary to that being that medium to large distances are required for sufficient isolation for speciation. The mechanisms of speciation that people typically talk about include these three right here. So we can divide them up in terms of allopatry, sympatry, and parapatry. Allopatric speciation has historically what people have been talking about in the literature for a very long time. The idea that species form because there's a barrier that develops between populations. There's some kind of barrier that becomes a physical isolator. And over time, as these populations remain separated by that barrier, they start to change evolutionarily and eventually evolve into different species. And allopatric speciation has played a really prominent forefront role in the discussion of evolution and speciation. Sympatry is quite the opposite case, where speciation can occur without physical isolation. And the idea here being that you can have populations that are completely overlapping in their distribution, and maybe they remain that way, but they can still, over time, become distinct at the species level. And perhaps there might be something like a slightly different food choice, something about their ecological niche that differs, even though they still have these overlapping distributions, that keeps them separate long enough that they eventually turn into different species. Parapatry, the third one I have listed there, is speciation with adjacent or only very small overlaps in distribution. Now, there's been a fair amount of research done over the last few decades that examines which of these mechanisms plays the most important role in speciation for marine organisms, especially in the tropical Pacific. But very little work has been done to date looking at seaweeds from this kind of perspective. There are some general patterns of biodiversity that we can see in marine areas, and there are several marine biodiversity hotspots that have been identified around the world. The largest of these is what's termed the Coral Triangle, and you can see it outlined in this area over here in the Western Pacific, Western Tropical Pacific. The Caribbean is another area, another marine tropical biodiversity hotspot, and then there are some regions of the Red Sea and the Indian Ocean that also qualify for these. It turns out that these are not stationary in geological time, and over the last 50 million years, these have shifted quite a bit around the globe, and they seem to mirror the tectonic events that are occurring on Earth. So researchers have put forward a series of hypotheses to explain these biodiversity patterns that we're seeing, and here I'm listing several of them. The first being the center of speciation, the idea being that you can have very large levels of biodiversity, high species numbers in areas where the speciation process is occurring very to a large degree. So there are a lot of species being generated in a particular area. And maybe that's one reason why we see, for example, a lot of species in the Coral Triangle. The second one I've listed there is the center of accumulation. And that's more or less the opposite pattern, whereby the assumption is that speciation is actually occurring in the peripheral areas at a much higher rate. And these species are being exported and accumulating in the Coral Triangle, for example. The third I have listed there is the center of overlap. And the idea there is that there may actually be several different areas of high biodiversity that are adjacent and in fact slightly overlapping with each other. In that area of overlap, you have all the species or a subset of species from both being added together, resulting in a very high level of biodiversity. Recently, Brian Bowen and his colleagues have put together the fourth hypothesis that I have there. It's termed the biodiversity feedback model. The idea being that there are actually several different mechanisms occurring here, that you might have different reasons for elevated biodiversity in different parts of the world, because some regions are exporting biodiversity and some are accumulating. And so there is some exchange between these peripheral areas and the central areas of speciation. Now, which or any of these actually apply when you're starting to talk about seaweeds, we really have no idea. There's been so little work done to date on the phylogeography of marine seaweeds in the tropical Pacific, at least at the level and the rate that we would need to understand these patterns, that we really can't say at this point. Let's talk a little bit about marine algal dispersal, because this plays a key role when you're thinking about how organisms transport from one area to the next, how you might have a connectivity among populations. So I'll go back to this assumption that I mentioned a few minutes ago, the idea that most marine species have a planktonic larval stage that can disperse well over short, medium, and or large distances. Let's look at what algae are actually capable of doing. So macroalgal propagules, so the dispersal units of seaweeds are typically pretty small. We can be talking about spores or gametes, the reproductive cells. These are single cells, and they often look something like this. Okay, so these are single cells, flagellated or not, and a single cell in an ocean basin really doesn't have a whole lot of chance of getting too far. This is so small, it could be on the order of a few microns, maybe 50 microns or so. It's going to be so overwhelmed by the forces of the water, it really doesn't have a chance to swim independently and get very far at all. It's thought that these are short-lived, on the order of hours or days that they're going to exist. They're really out there for the express purpose of finding a mate in the case of gametes, or finding an appropriate substrate to settle on, things like spores. Okay, so they have a very short lifespan, and they're really not going to get too far. Some of these are non-motile. The ones that are not have very limited motility. They're just not going to be able to travel the great distances between, for example, island sets in the Pacific Ocean. So what else is getting around? I mean, we do know that there are a number of seaweeds that have a lot of affinity amongst the different islands that we see in the Pacific. Why is there a relationship among them? Long distance dispersal is probably playing a really key role in the evolution of the species that we see across the Pacific. And there are lots of different ways that you can have bits of seaweeds travel and establish new populations. A good example would be the rafting of large fronds, and I'm showing you here a photo of some kelp down here. There are large kelp beds that can become entrained in ocean currents, and travel as that entire bed from one region to another. They can go on the largest ocean currents. When they do that, they're often carrying other organisms with them because they are a habitat unto themselves. So there can be lots of attached animals and other algae as well living amongst these rafts. We can also have dispersal of filamentous life history stages. Here's an example right here. Asparagopsis taxiformis is one of the algae that I'll be talking about in some detail today, and it has an alternate life history stage from the part that we typically see, and this is what it looks like right here. This is a sporophyte phase, and it's small and bushy and filamentous, and it likes to grow in fairly thick clumps, and quite often when it grows to a certain density, it'll detach from the substrate, and it can be carried off and settle into a new region. It's a sporophyte, so producing spores then, it has the capability to generate a lot of new plants wherever it settles. The final way is dispersal of fragments, and what I mean by that is that most of these algae, these seaweeds, have the capability to be broken up into smaller bits, and in many cases these smaller bits are capable of developing into whole new adult plants. So sometimes all you need is a small bit that can be broken off by natural or by anthropogenic forces. And if these are traveling by wave motion or being carried by humans, then it'd be pretty straightforward to the establishment of new species in new areas. Okay, so most of the patterns that I'm going to be showing you today when we're talking about tropical Pacific marine algal phylogeography have to do with DNA data. And using DNA to get information about the evolutionary history of these organisms has really turned around our whole perspective of these organisms. It has been such a wonderful application to really getting a handle on how these things are moving around, where they've come from, how closely they actually are related to things that look very similar to them. And so I've put up here that the advent of molecular analyses also equaled the advent of algal phylogeography. And that's because I believe most of the patterns that we were inferring prior to this were pretty easily overturned. You'll see what I mean as we walk through some of these examples. So these techniques that we're using, that we do very commonly in the lab, involve going out and collecting large numbers of specimens. We try to get representative specimens, either here in Hawaii from lots of different islands, different sides of the islands, different populations. And as many cases as we can, we try to compare these to other places around the Pacific or other regions of the world if they're distributed pretty broadly. There are two different ways that I'm going to be showing you the DNA sequence data, and I've got them listed up here. So over on the left-hand side is a phylogenetic tree, and what this is a depiction of the relatedness between the different specimens that we've analyzed. So over on the right-hand side here, there's a list of names, and these are just the sample names. So maybe we had an alga that we collected from a particular beach park, and we'll be labeling it that way at these tips of the tree. These lines that are going out from those samples indicate how closely they are connected. That is, how similar their DNA sequences are to these other specimens that we've analyzed. So in this particular case, you can see that these 4 specimens up here at the top are more closely related to each other than either of these is down here to those. Okay, so it's a way of showing you evolutionary distance. The other kind of technique I'll be showing you are what we term haplotype networks, and that uses the same kind of data. It's also DNA sequence data, but what it's doing is analyzing a little bit differently and depicting it differently. So it looks kind of messy, but what you end up with here is a network. What it's doing here is taking each unique sequence, and that's termed a haplotype, and showing you the connections between it. So perhaps, for example, down here, we have a series of sequences that we've collected from samples from Maui, Lanai, and Molokai. They're identical to each other, and so they group together. They're a single haplotype. And you can see how closely related that haplotype is, say, to this one over here. These little open circles here indicate the number of nucleotide base changes between the sequences. So the more circles you have between them, the more distant the samples are from each other. So it's just a different way of depicting the data. So what I'd like to do is run through some examples. Some are projects that we've worked on in the lab, and others that I've obtained from the literature, just to bring together a series of examples and hopefully glean some patterns at the end. I'm going to start off with the red algae, because the reds are the most speciose group that we have. We have, at least 400 species here in the Hawaiian Islands alone as members of the red algae. This first example here, Amansia glomerata, is very widely distributed and very common. It's very simple to find in almost any beach park that you can go to around here. This is an upright foliose, bladed red alga. It very commonly has other organisms growing on it and amongst it, so it can be a little bit dirty, but it's extraordinarily common. This alga here was described from the Hawaiian Islands back in the 1800s, but it has been since located in a number of places around the world. This map that I'm showing you here is downloaded from the Global Biodiversity Information Forum, and it's giving you reported distributions of this species and others that I will show you. So the specimens that we studied were all from the Hawaiian Islands, but you can see there are other records from other places in the Pacific, including Australia and Japan. So we were doing a large-scale biodiversity survey of the red algae of the Hawaiian Islands a few years ago now. And as part of that, we collected a number of samples of this alga from the main Hawaiian Islands. And we also were able to obtain some specimens from Bishop Museum to use in these analyses as well, to increase the distributional patterns that we can see here. What we can see in these analyses is that we didn't have one network, but we actually had 3 different networks that came out of these samples of what we thought were the same species. And typically what this is meant or interpreted as is species boundaries. So with this 95% connection limit with 3 different networks falling out, people would start to investigate this as the possibility of being 3 different species, which is kind of interesting. If you start to look at these different types in terms of a map, so here's a map of the Hawaiian Islands, and I've indicated a pie chart for each of the islands, and it's broken up by the different kinds of sequences that we found for each. You can see that there's one lineage here, labeled lineage 1, that's in white, and it's showing up in some of the main Hawaiian Islands. Lineage 2 in gray, that's up in the northwestern Hawaiian Islands, and then lineage 3 is pretty common around the main Hawaiian Islands. So you can see that the northwestern Hawaiian Islands are distinct from the main Hawaiian Islands. There's a different sequence type that we're finding in these 2 locations, which is kind of interesting. Are they really the same species? This became the big question for us when we started to look at how different they were from each other. This alga is what we call phenotypically plastic, which means that there's a large range in the morphology that you can find that you would expect to find for the species. They can look very different from each other and still be considered this species. So where do you draw the line on what one species is versus the other? The morphological variation we were looking at wasn't consistent, so that all the northwestern Hawaiian Island specimens looked a certain way and the main Hawaiian Islands looked a different way. Rather, they were quite mixed up with each other. The sequence divergence that we found as well was sort of in between what you would expect for within a single species versus between 2 species. So this one, it was kind of left up in the air. We don't really know if these are different species or not. It's quite possible that these are species in formation, that we're looking at evolution in action essentially here, where these are beginning to diverge from one another, but haven't completely yet. And so we see an intermediate level of divergence. I also wanted to say that for this project, we were lucky in being able to sequence the type specimen for the species. So this is the one specimen in the world that anchors the concept of this species. And being able to obtain a sequence for that species was critical, because what it means is that if, in fact, these are described down the road as 3 different species, this one up here is the true Amansia glomerata, okay? And then these 2 down here would receive different names. Okay, Asparagopsis taxiformis is another alga that we've been interested in for a long time. This is Limu kohu, and it's an extremely abundant and very important, culturally important alga here in the Hawaiian Islands. It is prized for its edibility here. And it has a very wide distribution, as you can see. There are locations around the world in the tropical and subtropical areas that have reports of Asparagopsis taxiformis, including, of course, the Hawaiian Islands. So there's been an assumption up until very recently that this is a single species, that it is a cosmopolitan species distributed very broadly and has a broad tolerance for environmental conditions. Back in 2007, there was a group out of Italy that published the first phylogeographic analysis of this species. And what they did was obtain a series of specimens from around the world, and they sequenced them, and they started to build these haplotype networks that I've described to you. to try to figure out if there were differences or if it's really all just the same thing. And what you can see is that they have it divided up into 4 different lineages. So they have lineage 1 up here. Each of these lineages has a map of the world beside it where you can see its distribution. They have lineage 2 over here, and you can see the number of differences amongst these 2 lineages. They have lineage 3 over here and lineage 4. Okay, and you can see that the number of base pair differences between these lineages, they're pretty well delimited. Now, when this study first came out, I thought it was really interesting because if you look carefully at it, you'll see that there are a series of specimens from Hawaii included in their analyses. And they show up in 3 out of 4 of these lineages, these global worldwide lineages that they've analyzed. I thought that was really interesting because I would have assumed that what we had here in Hawaii was just one thing. Because we're so isolated, my initial assumption would have been that, yeah, there was probably just a single colonization event and what we have here in Hawaii represents a single lineage. That's obviously not the case. Okay, so that enabled us to begin our own work looking in more detail here in the Hawaiian Islands of what's going on with Asparagopsis taxiformis. So we just increased the number of specimens that we looked at for this alga around the islands, and we had a number that we obtained from the main Hawaiian Islands and then a number of specimens, both from cruises and from Bishop Museum for the northwestern Hawaiian Islands. What I've done here is labeled the 4 lineages that Andriakis et al. Had described on the previous slide. And you can see we found very much the same pattern that they did, but in a little bit more detail. So you can see that we have lineage 1 in yellow. That's very common throughout the main Hawaiian Islands, and it's the only lineage that we came up with on some of the islands. Lineage 2 in green is really the only lineage that we see in the northwestern Hawaiian Islands, except for here in Nihoa. But for the more distant northwestern Hawaiian Islands, everything seems to be lineage 2, at least so far. That's important because it might indicate that this was the initial original lineage, if you consider that these islands are very protected and undisturbed relative to the main Hawaiian Islands. Lineage 3 we do not have, as far as we know, here in Hawaii. And then lineage 4, we came up with a few specimens on the island of Oahu. These included locations like Ala Moana Beach Park and areas in Waikiki. And when we start to look at the dates of when these lineages appeared, it seems like this may be a very recent arrival, just in the 1990s, according to specimens that we have at Bishop Museum. So this is a lineage that has probably come in pretty recently from somewhere else. Now, interestingly, people are studying this species in a lot of detail because we're getting a really good database of reference specimens around the world. And this is a paper that just came out last year looking at the species in more detail in New Caledonia and other parts of islands off of New Zealand and Australia. They titled their paper, "The More We Search, the More We Find, Discovery of a New Lineage and a New Species Complex in the Genus Asparagopsis." And I think it's really apropos because it's true. The more we search, the more we are finding. Essentially, the more specimens you add into these analyses, the more complexity you end up uncovering. None of the samples, none of the species that we've been investigating on the assumption that they're cosmopolitan have turned out to be that way. They turn out to be interesting complexes of things. Okay, so here, this is a more complex haplotype network, but essentially all they're doing is adding in this one lineage up here, lineage 5, and it is found in New Caledonia, Kermadec Islands, French Polynesia, Lord Howe Island off of Australia. There's an additional lineage that's been discovered since then and is currently in review. So indeed, the more that we search, the more we find. I wanted to talk a little bit about Portieria hornemanii. This is a common species here in the Hawaiian Islands, and as it turns out, it's pretty broadly distributed in other regions as well. The assumption, up until very recently, has been that Portieria consists of a single, widely distributed species in the Indo-West Pacific. There's only one name that people apply to collections that look like this, hornemanii. Okay, so the first study that was done on this focused on a region in the Philippines where they were looking at island-specific patterns of this taxon. This shows you the global distribution of the taxon. Their study focused in here in this region outlined in red. So they did much the same kind of study. They went out and they collected hundreds of specimens of this alga around the Philippines. They sequenced them, They analyzed those sequence data and tried to look at the patterns. How do they fall out into different groupings? And what they found is that there were actually 21 species that seemed to be delimited based solely on the specimens that they had from the Philippines, which is pretty incredible. I mean, if in fact they do represent biological species, we're not entirely certain at this point. They haven't gone so far as to name all of these. But using species delimitation models, they came up with this approximation. This work is ongoing, and there's a really interesting group called the Porchiaria Evolution Consortium, which is embracing this taxon as a model organism for the study of seaweed phylogeography and the untangling of cryptic species. This is headed up by Frederick Leliart in Ghent, in Belgium. And this is his data that I'm showing you, but it does include samples from Hawaii that we have provided, and we're members of this evolution consortium. Up here you can see this is a phylogenetic tree turned on its side, and you don't need to read any of it. All I want you to see is that there's many, many specimens from throughout the distributional range of this species that people have been sending in and having sequenced and added into these analyses. The specimens from Hawaii fall out into this little group right here. What they've done is apply a whole series of interesting species delimitation models, so ways that you can tease apart the DNA sequence variation, trying to determine where that boundary is between what you would expect for within a species and between species, and use that as a means for delimiting these different species. So what these analyses are suggesting is that Porchiaria is composed of many, many more species than previously recognized. If this is a single species, it all goes by Porchiaria hornemannii, and now we're recognizing all these different species in these different regions. You can see how the diversity is multiplying incredibly based on these methods. Spiridia filamentosa is a really common red alga around the Hawaiian Islands as well, and it has a very broad distribution, like most of the taxa that I'm talking about today. We choose these species because they have these broad patterns of diversity, and what we want to see is, are they in fact as widely distributed as it appears they are, or are they more restricted in distribution? This species has been studied for a fairly long time. Some of the first phylogeographic work came out in the early 2000s, where it first became evident that there are at least two major clades or groups of these specimens. The top one being termed the Pacific Ocean clade because it contains specimens from the Pacific Ocean, except for Greece, of course, up here. And then this bottom group that had a whole variety of things that fell into the species concept of Spiridia filamentosa, including things from the Atlantic Ocean, the Indian Ocean, and the Mediterranean. Okay, so I had a master's student that joined the lab some years ago, and she was really interested in looking at the diversity of Spiridia filamentosa around the Hawaiian Islands, but she had an application for this as well, which is, I think, what made her master's thesis really interesting to me. She was fundamentally interested in trying to use this species for restoration of seagrass beds here, actually in Honolulu Bay on Oahu. And so what she wanted to do was be able to pick certain haplotypes or certain types of this Spiridia filamentosa that could be outplanted. And so after they removed some invasive algae and outplanting the Spiridia, this alga would trap the sediment. It's very good at that. It has these small, fine branchy filaments, and build up the habitat necessary for the seagrass to be restored. So she wanted to use it for this purpose. In order to do that, she wanted to know whether there was a native haplotype to this region versus going out and just collecting Spiridia from some other part of the Hawaiian Islands. Maybe it's something that didn't even belong in the area. So she really wanted to tease this apart before getting into the restoration. So she ran a whole series of DNA sequence analyses based on specimens that she collected around the main Hawaiian Islands. And I won't go through all the patterns here, but just note that we're seeing something similar here to what we've seen in almost every example so far, that it's not just one thing, that here in Hawaii alone we have 6 different genetic clusters of this species. And they're allied with things that you find in lots of different parts around the world. And so it appears that things are actually moving around a fair amount. That the seaweeds that we have here in Hawaii, even if they're things that have been recognized here for a very long time, decades or hundreds of years, that's not necessarily the same thing, that there may have been other lineages of this that have come in much more recently. talk briefly about Bostrichia because it's an example of a different kind of study that's been done. There are a couple of species in this red algal genus, Bostrichia, that are commonly identified, and I'll be talking about this region of the world right here with specimens. There are, I have a colleague who did some work in the late 2000s who was looking at DNA sequence diversity of specimens of this. What he found is that there are 7 lineages that he uncovered with the DNA sequence data that didn't correspond very well to the 2 taxonomic names that are being applied. Okay, so it's sort of a mess. 2 names, 7 genetic lineages, what matches what? They're not adding up quite right. There's a lot of mixing going on there. What I liked about this study, though, in particular, was that he actually tried to cross these to find out whether they are distinct species or not. So he did essentially mating experiments with these 2, with all these different lineages, and tried to figure out, okay, if we try to mate lineage A with lineage B, what happens? Are they species under the biological species concept or not? Most of these lineages were reproductively isolated from each other, so it looks like there is much greater diversity than just the 2 names that have been put out there so far for that group of specimens. Some of these being very localized, only found in one or a very defined region, and some of them much more widespread. Okay, so moving on from the red algae, I'll present a single example of the green algae and then talk briefly about the browns before wrapping up. So the green algal example is Halimeda discoidea, a very common green alga. And you can see its distribution here. So again, essentially pan-tropical, many regions around the world. So this alga became the basis of a PhD thesis that was conducted by Jeroen Verbruggen. And he published part of his data in 2005, what I'm showing you here. And what he did here was similar to what I've talked about before, analyzed DNA sequence data for specimens from a very wide geographical range. But he analyzed a couple of different markers here. So he has a nuclear marker over here, so something that's coded in the nucleus, and then he has something that's coded in the chloroplast. So that's a different genome within that same alga. And what you can see is that there's a different pattern, a different signal entirely that's coming out from these 2 different sources within the one alga. And so it's not uncommon to find this, but it's a reminder that, you know, people that are studying these kinds of patterns need to be cognizant that you do sometimes uncover slightly different patterns depending on the kind of data that you're using. What he found is that the chloroplast markers showed much greater separation between Hawaii and French Polynesia in the Philippines than the nuclear data did, so a different pattern. And similarly, the nuclear data showed much greater diversity in samples than you find from the Philippines. Okay, so switching to the brown algae, I'll show you a couple of examples here. And these are both species that are very widespread, and they're also pretty uncharismatic, I would say, in their morphology. And what I'm often finding is that these are the kinds of species in which you find a lot of diversity that's been overlooked, because they just don't have a huge number of really distinctive taxonomic characteristics to go on to describe them as different species. And so, you know, coming across things like this when you're diving, it's somewhat unremarkable compared to a lot of the really beautiful red seaweeds that you see out there. This is a brown crust, essentially, and unless you look at it under a microscope, it doesn't have a whole lot of interesting morphology going for it. It's very broadly distributed, though. Lobophora species are known pantropically around the world. I want to show you the results of one study that just came out very recently, last year, by Vieira et al. They focused on specimens from New Caledonia. Okay, so they were just looking in this one island area and just using this one particular brown algal taxon. They looked in the literature when they started to analyze their data and found that there were 4 names that had been applied to this genus, Lobophora. When they did their analyses based on DNA sequence data, though, what they found is that there were anywhere between 31 and 39 distinct units that could be delimited. These are labeled up here as MOTUs. That stands for Molecular Operational Taxonomic Units, and all it means is that the sequences are clustering in that way, and you can define them as a distinct group from one another. So they applied species delimitation techniques to look at their DNA sequence data sets, and that's why you see the variation in the number of MOTUs that they document. But by all methods that they looked at, there are many, many more units out there than people previously recognized. They went further in this study than we're typically finding in the literature these days. That is, they went ahead and described 10 new species based on these analyses, plus very detailed morphology and anatomical analyses of these specimens. Then this last example is Culpamenia sinuosa. Again, a very common brown alga, very common here in Hawaii, as well as many regions in the tropical Pacific. What you can see here is some of the variation in morphology in this alga. This all goes by the same name, Culpamenia sinuosa, but there's a fair amount of variation in what they look like. And that's its distribution, okay? So very, very broadly distributed. Okay, so in this study that came out in 2013, they analyzed a series of specimens from as many places as they could sample throughout the range. And you see that it is divided up into a series of groups. They have 3 broad groups that are identified, 1, 2, and 3. And within those groups, even, there can be a fair amount of variation. 2 of these overlapped in distribution with each other. And they attributed the distributions as combined effects of anthropogenic dispersal and ancient biogeographic patterns, which I think sums it up pretty nicely for a lot of the things that we're looking at here today, that humans are probably involved in the distributions that we're seeing, that there has been so much traffic, both inadvertent and perhaps in some cases purposeful plantings of things, that the distributional patterns that we're trying to untangle are very much overwritten in a lot of cases by more recent events. So to try to summarize some of the things that we've looked at this afternoon, there's increasing evidence for high degrees of speciation in tropical seaweeds, even though they have poorly dispersing propagules. Things like Porchieria, where they found 20-some species in the Philippines alone, where there's just a single name that's applied across the tropical Pacific for that. And Lobafra, this uncharismatic brown alga, which has many, many units within it when you start to look in detail at the DNA sequence data. We know at this point that morphology-based taxonomy is failing to provide accurate estimates of algal biodiversity. This is something that phycologists or people who study algae have been slowly recognizing and acknowledging over the last decade or so, that we cannot rely solely on taxonomic keys. We cannot only sit at the microscope. We cannot only use the guidebooks that take us out and try to identify things in the field. They are inaccurate. You absolutely need to be looking at other characters in order to understand the diversity of these species. They, in many cases, just have so few characters, or they just haven't been acknowledged as useful taxonomic characters yet, that we need to look at the molecules and then work back from there to say, aha, it's a different clade. Let's start to look in detail for things in the anatomy we haven't used before to identify. So coming out of that, we know that cryptic species discovery is common. We're recognizing that there are lots of species that we can't really tell apart morphologically. It doesn't mean they aren't species. Maybe we don't know exactly what the appropriate characteristics are yet to tell them apart, but the DNA molecules are indicating very clearly that they're on separate evolutionary trajectories. They're not the same species. This discovery of cryptic species discovery, it yields many more species than previously recognized, but it also retools the scope of phylogeographic analysis. What I mean by that is that I stood up here today and wanted to talk to you about phylogeographic patterns of tropical marine seaweeds, but the truth is, every time we try to do this, what we typically discover is that it's not a species, it's a whole series of species. And if you want to look at phylogeography, you need to get below that species level. So it's retooling that scope. Increased sampling yields increased lineage diversity. For example, the case of Asparagopsis, where every time another group of researchers goes and looks in detail at a new region of its distribution, they're finding new entire lineages that we didn't know about before. This highlights the need for the study of algal specimens at a fine geographical scale. It's no longer adequate to do the kinds of studies that we used to do 10 or 15 years ago, where we would gather a few specimens from a few places around the world and try to infer phylogeographic patterns. There's just so much going on, so many patterns to untangle, so many genetic lineages within these taxa that we need to cover these at much finer geographical scales to start to see what's happening. And then also, unraveling phylogeographic patterns is becoming increasingly difficult with human-mediated dispersal of marine organisms, whether intentional or not. We know for a fact that these are being distributed by many different activities. There are ships that carry them. They can grow on hulls. They can grow in ballast water in some cases. They can be used as packing material for shellfish. There are lots of ways that seaweeds are being distributed around the globe, and that really is influencing the patterns that we're trying to uncover. So future research, we've only got a few species that have been looked at in a lot of detail so far. Many more examples would be required before we can start to summarize these patterns and get back to the introductory material that I talked to you about today. The idea of trying to look at these biogeographic provinces and trying to look at centers of diversity and trying to infer whether speciation is higher in certain areas than others for seaweeds. We just don't know yet. How do we get there? I think we need large cooperative projects, and the Portuaria Evolution Consortium is a great model for how this could be done. It'd be lovely to be able to answer questions like, do the Hawaiian Islands export marine algal biodiversity? We don't know at this point. So broad-scale biogeographic patterns can be called into question when molecular techniques are applied to the study of algal distributions. It pulls it back from an examination of phylogeography and indicates the level of cryptic species variation that we have that we need to look into in more detail. So I'd like to end by thanking a few organizations and people who've been involved in this work. My colleague Joe Zuccarello in Wellington, New Zealand, is a co-author for a book chapter with me on this topic that we submitted relatively recently. The work that's been done in my lab has been funded by the National Science Foundation, and there's been a whole series of researchers in the lab working on this topic, but I'd especially like to thank Kim Conklin and Dr. Akira Kurihara, who was a postdoc in the lab and worked very heavily on the red algae for a number of years. And lastly, I'd like to thank the Hanauma Bay Education Program for having me today, and thank you very much for your attention.
Any questions?
Your talk suggests molecular genetics as a way of determining a broad diversity of a single species. But does it suggest time? Say the natural distribution. Can you speak of the time element?
Yeah, there are some techniques you can apply to DNA sequences to put more definitive dates on things, especially going back pretty far geologically. Yeah. You can do that, and we haven't done it so much here in the Hawaiian Islands because it's all really pretty recent, actually. There are some other ways that you can do it. Just looking at the variation between the sequences, and in other cases, looking at when certain lineages arrive. So if there is a record, like for Asparagopsis, looking at the specimens in Bishop Museum, we sequenced everything we could for Asparagopsis from Bishop. And what we found is that there was one lineage that didn't appear in those collections until the 90s. So that's a way to at least infer more recent distribution patterns, where that one probably came in more recently. It wasn't picked up in the older collections at all.
Any thoughts on the plasticity of a species? I'm saying that in the big sense of the way they've previously been delineated, and the niche as far as the development of some of this plasticity. I say that in a way that
I think so.
Someplace running around my head.
Yeah. So a lot of the variation that we see in these seaweeds that are known to have a whole variety of morphologies associated with them, some of that can be explained by the environment in which they live. So there are some that will grow longer and weedier looking when they're not in higher wave action because they're not being battered by the waves and they're not being broken off. And the converse would be true in higher wave action environments where they're going to be more compact and robust. So they're certainly influenced by the environment in which they live. And trying to tease that kind of variation apart from species level, like ingrained genetic variation, I guess, is the trick in figuring out which characters are going to be valid for describing species and telling them apart from each other. What's it capable of doing if you take a species and put it in a different environment versus what are the things it's going to do no matter what? Yeah, that's a challenge.
Okay.
Yeah. Yeah.
I've always been good at coming up with challenges that were not yet possible with technology.
Yeah. So how do you account for it? You can do experiments, but I think more commonly what we try to do is collect an alga from a variety of different habitats and look at it that way. So if you know from the DNA sequence that it corresponds to a certain entity, do you have some in that group that are found in high or low wave action or whatever the habitat difference is, and do you still see that difference or not in that group? So that's one way to tackle the question.
But it hasn't said that much.
We do it to the degree we can with the level of sampling that we have in different environments. Yeah.
I was just wondering, are there any similar studies being done with temperate algae as far as distribution and genetic differences between algae?
Yeah, definitely. And there are some pretty notorious species in terms of invasive taxa in temperate environments as well that are being focused on in a fair amount of detail.
And human impact on that distribution also in temperate algae. Area they're looking at more recent distribution caused by human distribution.
Yeah, I think there are probably not that many places on Earth that haven't been touched by human dispersal at this point.
Any other questions? Thank you so much.
Thank you very much.